Happy to have the chance to talk with you today and answer any questions. As Madeline said, I'm a professor at MIT. I also have a different role. I'm a vice provost for faculty, and I have a lab. And my research lab examines how we can use charged polymer systems to make materials that can deliver drugs. So I'm going to talk about one particular set of materials which are nanoparticles, but I'll describe the big picture as well. The method that I use is a very simple one. You take a substrate, which can be any sort of solid surface. And if that solid surface is charged, let's say it is negatively charged, you can immerse it into a dilute aqueous solution that contains a polymer of opposite charge. That polymer chain will adsorb along the surface of the substrate until ultimately that surface charge gets reversed. And then anything else that comes toward it will be repelled. So you have no more deposition, and you have this monolayer of polymer that has adsorbed. You can rinse the substrate to remove anything that's not truly adsorbed, and then put it in another dilute aqueous solution that has a polymer of the opposite charge. And you adsorb that until you get surface charge reversal. These adsorptions can take place pretty quickly, and you can rapidly build up these thin films. And these thin films can contain a large number of different things in them as long as you follow the rule that you are alternating charge as you build. The films are extremely thin. They're somewhere between a few nanometers in thickness to a few tens of nanometers in thickness. And when we began looking at how we might apply this approach to drug delivery, we were attracted to the fact that we could incorporate a large number of natural drug materials. These are natural polymers like proteins, RNA and DNA, and even some of the small molecule drugs have charge to them. So we can incorporate these different kinds of components into the thin film and incorporate something that might degrade to help release it. So in that work in our lab, over the past several years, we've been looking at uh, layering very large scaffolds that can be then incorporated for tissue engineering. For example, in this picture, we were incorporating a protein known as BMP2, bone morphogenetic protein 2, which recruits the stem cells from the bone marrow to the site and enables those stem cells to become bone cells. So here you can see bone in micro CT where there's a defect, and we have covered it with our film, which you cannot see in micro CT. But over time, what you can see is that the bone begins to regenerate over time. So we had projects in orthopedic applications like bone regeneration. We also coded things that were a little bit smaller, like the micro mesh that you can see in a bandage. And in these bandage applications, we were incorporating in the layers siRNA or silencing RNA that would shut down some of the genes that are dysregulated in wound healing, for example, for diabetic wounds, helping those diabetic ulcers to close. So we had projects that looked at siRNA release. And we went down to even smaller length scales by layering micro needles, patches that had tiny needles that only penetrate the very top dermis layer of the skin. And we coated those with DNA and used that as a way to create DNA vaccines. So now I'm at the Koch Institute of Integrative Cancer Research, where we work with biologists to try and solve problems to address cancer. And in that work, we were very interested in being able to design a system that can deliver drugs specifically to tumor cells without engaging healthy cells, in part because we're typically trying to deliver a drug that will kill those cells, and we want to only attack the cancer cells. But we were also interested in using our methodology because a lot of tumors, a lot of cancers are resistant to chemotherapy drugs. So the way that we did this was we started with a nanoparticle. Nanoparticles are small. They can navigate through the bloodstream, circulate, and ultimately find their way into the tumor tissue. and we needed something that small. So the idea was we can take a nanoparticle like a liposome or a polymeric nanoparticle. We can design it so that it has a net negative charge. And we can stuff inside that nanoparticle something like a chemotherapy drug that is going to kill the tumor cells. If we design it for negative charge, we can absorb a positively charged polymer on top. And that is often a peptide, a polypeptide. And then we can absorb negatively charged nucleic acids like siRNA. And in this case, we're choosing an siRNA that will silence the genes that enable tumors to actually defend themselves against the chemotherapy drug. And this is how we're really trying to address resistant cancer types. Then we coat that with a positively charged polymer again. So we essentially have a sandwich where we have this positively charged material, then the siRNA, and another positively charged material on the other side. This protects the siRNA from being degraded by the proteases and RNases that are present in the bloodstream. But if we actually introduce the particle with just that plus charge on the top surface, it's going to absorb a large number of plasma proteins. And a lot of you study chemical biology and chemistry, and you know that a lot of those are negatively charged proteins. What happens when you get a large agglomeration of proteins on these nanoparticle surfaces is they get rapidly eliminated by the immune cells that are present in the bloodstream, like monocytes. So we won't have a nanoparticle that is going to circulate for a long time if we just deliver it in that fashion. We need one more layer, and that's another negatively charged polymer. Negative charge is useful to us because other cells usually have negatively charged glycans on their surface, which means that they are going to be repelled from interactions with this nanoparticle. And that lowers the chance that we're going to interact with healthy cells. So we want something that is going to be very hydrated as well, because that means it's going to be less likely to absorb proteins because there's a barrier, a penalty for proteins coming along and displacing these water molecules that thermodynamically are so happy associated with our layer. So there are a number of ways in which this becomes a stealth layer, a layer that causes the nanoparticle to go through the bloodstream without being rapidly recognized by immune cells and eliminated. And this is what these nanoparticles look like. On the right, you can see a polymer nanoparticle, a degradable polymer. We can coat that with 4 layers, and we get a kind of shell. And that shell, that transparent shell, is something like 20 to 30 nanometers. You can see the liposome in the other corner here, before and after coating. And you can see again that we're sort of coating these systems. We can also coat other kinds of vesicles and lipid nanoparticles. And, you know, I like to think about this as a way of delivering drugs in sequence. And so I often would use this jawbreaker to describe it to others and talking about the science, that you have this Wonka gobstopper. I don't know if any of you remember that, or these jawbreakers where the different flavors or layers are exposed as the jawbreaker dissolves. So I mentioned that we use this outer layer, and that outer layer ends up being really important for the work that we do. And here you can see some of our earlier work in which we looked at that final layer as hyaluronic acid, which is this high molecular weight polysaccharide that is native in our bodies, in our tissue. It turns out that hyaluronic acid is very highly hydrated, and it is a great stealth layer for that reason. But it also has a binding affinity for a receptor, a protein receptor on cancer cells, CD44. It's highly overexpressed on these cancer cells. And for that reason, these nanoparticles may avoid healthy cells fairly well, but they are able to engage with cancer cells, as you can see by this image here, where the red nanoparticles are associated in the cell. When we introduce the nanoparticle into a mouse that has tumors on its back, we can introduce the nanoparticle through the tail vein, let it flow through the bloodstream, and it begins to accumulate. And here in this image in histology, we stained the nanoparticles, they're green, and CD44 is used to stain the tumor tissue. So all of that orange region is where the two are overlapping. You can also see that the nanoparticles penetrate through these outer layers. These are protective stromal layers that protect the cancer. We're able to get through them and to the cancer itself. So when we first started testing this technology, we decided to look at a more realistic case of cancer. And we decided to examine non-small cell lung cancer, which is a very aggressive form. It also has some unique genetic modifications, and we can use siRNA to target those genetic changes. Um, Here we're looking at our first experiment, which is to see where the nanoparticle goes in healthy mice. It has a long half-life. It lights the entire mouse up when you inject it in the tail vein, but eventually it just gets eliminated and shows up in the liver briefly before it's all gone. But when we look at mice that have this native form of lung cancer in their lungs, we see that there's a huge amount of accumulation of the nanoparticle in the lungs compared to the healthy animal. In fact, there's 40 to 50-fold more nanoparticles in the lungs. And when we stain those lungs to see if the CD44 receptor is present, we see that indeed the cancer does overexpress CD44, which is why we're seeing that targeting. So we can then load up this nanoparticle with an siRNA against a really common oncogene in non-small cell lung cancer, KRAS, and a microRNA that replaces the function of another gene that is lost and helps to protect cells from cancer. So we're trying to really address those genetic modifications in the cancer. And we're going to put inside of the nanoparticle cisplatin, which is a common drug that is used, but isn't often successful in aggressive forms of the lung cancer. Here we're looking at results from an animal study, and we used red tracing to show the tumor because we didn't have labeling of the tumor in this model. And what you can see is the animal that is untreated had large tumors. If we just introduce the RNA, or we just introduce the cisplatin alone with the nanoparticle, we see there's some reduction. We also see that there's some improvement in survivability when we look at how long the mice survive. But when we have a combination of both the RNA and the cisplatin, we get something like 40% survival and much smaller tumor growth. So we were very excited about this approach, and we've been using it for other cancers like lymphoma and glioblastoma. But we've been really interested in trying to address ovarian cancer. But ovarian cancer presents a few challenges. For one thing, it doesn't have this very unique signature genetic modification like the KRAS that I described before, or like HER2 in breast cancer. So we can't use the same sort of personalized medicine approach, and delivering siRNA becomes less effective. The other thing is that ovarian cancer is really subtle. The symptoms are very minor in the beginning, and for that reason, it's usually detected fairly late in the third or fourth stage. So we really need mechanisms for detecting it early, which is work that we're beginning to do with some of my colleagues here at MIT. However, once it is discovered at this third or fourth stage, typically patients respond to chemotherapy, but after the chemotherapy is done and there's some sign of recovery, a few years later, about 85% of those patients experience a recurrence of the cancer, usually in a really resistant form to the original therapy. So this is awful because we have a huge amount of that patient survival loss happening in this recurrent form. It's a great chance to use immunotherapies where we train the immune response so that it sees that cancer coming back and addresses it before it can grow. But unfortunately, ovarian cancer is not responsive to common immunotherapies. The reason for that is that common immunotherapies usually treat the immune cells that are already present in the tumor. And in ovarian cancer, there are very few of those immune cells present. So we decided that we would try to address this by first understanding which nanoparticles are going to have a high affinity or stickiness to the ovarian cancer cells. And in doing this, we decided we'd make a library. We look at a bunch of different outer layers, and we did this by taking liposomes that were negatively charged. We layered positively charged polymer on them, and then we would look at different negatively charged polymers and determine which of these had a high affinity to ovarian cancer cells over healthy cells. We actually found that there were three outer layers that had a huge affinity for ovarian cancer cells, but didn't seem to interact a lot with other cells. One of them we've just been talking about, and that's hyaluronic acid. If you label the nanoparticle green, here we have fluorescent nanoparticles, and you see where they are in the cell, they go inside the cell into these endosomal compartments. And then they ultimately make their way out through an endosomal escape mechanism. That means that you can deliver siRNA and chemotherapy drugs, all the things that work on the cell on the inside. But we also found poly-L-glutamic acid coated nanoparticles tend to stick on the outside of these cells and just stay there for hours and hours. So we found that interesting because they never go inside. And they're also in between polymers like polyl-aspartic acid, where you got nanoparticles inside, but very slowly, and they ended up in different kinds of compartments in the cell. So in short, we found that the outer layer navigates this nanoparticle directly to ovarian cancer cells and in a selective fashion, but it also determines where the nanoparticle goes within that cell. And two of them were really interesting for a therapy approach. Hyaluronic acid, as I described before, and polyl-glutamic acid. Both of these formulations are really faithful to where the tumor is. So because ovarian cancer spreads throughout the abdominal cavity or the peritoneal cavity, if you inject it using injection that is intraperitoneal directly into that abdominal cavity, about 85 to 90% of the nanoparticles go to tumor. Here you can see labeled tumor. It has a luciferase signal. This nanoparticle has an IR signal, and you can see that these are directly overlaid on each other. So the nanoparticle is where the tumor is. Even when we see metastases, tumors that grow in other parts of the body, here we see a met to the intestine, and we see that our nanoparticle also goes to that met. So we have a really nice way of decorating ovarian cancer cells with these nanoparticles. And as I mentioned before, the polyl-glutamic acid formulation tends to stick on the outside of the nanoparticle. And in talking to my collaborator, Daryl Irvin, who's a bioengineer and works in immunology, he said, you know, a great molecule to present on the outside of cells is a cytokine. A cytokine is a protein that actually recruits immune cells to the site and then activates other cells. We thought this was really interesting. IL-12 is one of these cytokines. It's very potent. And when it is active, when it actually interacts with an immune cell, it produces interferon gamma. That's a really major inflammatory molecule that actually recruits additional immune cells to the site. So even though there are only a few immune cells present in ovarian cancer, we might be able to stimulate those immune cells to generate interferon gamma and bring more immune cells there. On top of that, IL-12 is active on T cells and NK cells. In particular, the CD8 positive T cells, these are the immune cells that attack the tumor once they are trained or informed on the antigen that the tumor is presenting. We really want these CD8 positive T cells present in the tumor space. IL-12 is really potent, but the flip side of it is it's really toxic. So the reason that you can't just administer IL-12 protein directly to a patient is that it gets throughout the bloodstream, and that causes the body to create its own autoimmune response. That can lead to extreme side effects, and that means it can even cause patient death, which was why it failed in clinical trials several years ago. So the idea here is that we can incorporate IL-12 into our nanoparticles that are then coated with polyl-glutamic acid. And in doing that, we will cause that nanoparticle to stick to the outside of a cancer cell, expose the IL-12 to an immune cell, and ultimately get that IL-12 mechanism going. And we use two different chemistries to attach the IL-12 to our nanoparticle. We took a liposome and we attached to the lipid in the liposome either a nickel hist tag, and so we used the histidine on the IL-12, or we used a malyamide cysteine and attached via the cysteine bond. And what we found was that when we use these LBL nanoparticles, in particular the formulation that had the malyamide bond, which is permanent, as opposed to the nickel his tag, which was one that breaks down over a short period, we found some differences in the numbers of T cells that were present in the tumors. And remember, the T cells are the immune cells we want to address the cancer. So here you can see us looking at the signatures for T cells, and we look in this square for a certain amount of CD45 marker and CD3 marker. And we see that they're not present for the unlayered nanoparticle that has IL-12. They're not present when we have IL-12 delivered directly to the cancer in the mouse because IL-12 is cleared so rapidly as a small molecule. It just gets in the bloodstream, but not in the tumor. We see that it's not present in the untreated mouse, but in our LBL nanoparticle, we see that it is highly infiltrated. In fact, if we count the number of T cells, we see that there are at least 8 to 10,000 more T cells present in the mice treated with our LBL nanoparticle, and that most of those are CD8 positive T cells. So if we dose the mice many times with large doses of our IL-12 nanoparticle, we find that we can get very, very strong survival. We were really excited by this result. But this was with a large dose of IL-12, and that really pushes the limits. And even if we incorporate the IL-12 in the nanoparticle, there's a risk to patients if it gets out. So we decided to go to lower doses, and we asked ourselves a different question. Checkpoint inhibitors, I'll skip ahead, they typically don't work in ovarian cancer because the immune cells aren't really present. But with our therapy, we're bringing immune cells to the site. And then the checkpoint inhibitors that are commonly used in most immunotherapies may be effective. So we decided to do a combination, a lower dose of IL-12 with an already known checkpoint inhibitor. We did 2 doses, and then we looked at how long the animals survived. And what we found was that our IL-12 nanoparticle with the malyamide linker actually gave us 100% survival for highly extended time periods. Here, we stopped the experiment at 200 days, but mice were still alive. We also found that along with this really exciting 100% survival, that when we took those surviving mice and introduced the tumor cells once again to the mice to mimic the recurrence of ovarian cancer, all of those mice survived again. So we've actually introduced an immune response that kills the cancer and that prevents new cancer from coming. We believe we understand how these nanoparticles work. They basically get embedded into the tumor. They stick there for a very long period of time, and they actually present IL-12 on the surfaces of these tumor cells, which then brings immune cells to the site. We're really excited about this. We've begun to understand more about the mechanism on a molecular level. And in doing that, we are understanding why polyl-glutamic acid is interesting and works. And it's because it's binding an amino acid transporter that is highly overexpressed in ovarian cancer cells. Now we've looked and we found some other cancers where this could actually be relevant, including forms of breast cancer, colon cancer, and head and neck cancer. Of course, we still have the formulation that goes inside. And as I mentioned before, we typically don't find that siRNA works so well for ovarian cancer, but there are combinations of small molecule inhibitors, like the one shown here, BCL1, XL1, and MCL1I. These are very potent small molecules that when you deliver them to the patient together can cause very negative outcomes. But in a nanoparticle, we can target it. And so the idea here is that we might be able to target the nanoparticle. Oh, wow. There we go. Sorry, it looks like I must have touched something like the crayon. What I'll do though is just say we found that this formulation is able to um, eliminate or greatly decrease ovarian cancer in the mice that we studied. And this is a newer piece of work, but we are now working on what we can do there. And if we can target immune cells, what else can we target? I mean, if we can target cancer cells, what else can we target? It would be interesting if we could directly target immune cells. And so we have Victoria, who's working on uh, putting different kinds of sugary polymers on the surfaces of our nanoparticles so that they might be guided to specific receptors that immune cells tend to have, which are based on carbohydrates. We have Victoria, and then we have Namita. Namita is working on coating lipid nanoparticles using the same kind of approach that I just described. She can actually change the degree to which these lipid nanoparticles are able to transfect cells and which cells they are effective in transfecting. And right now we're using her technology to target hematological cells like hematopoietic stem cells, with the idea that we might be able to address sickle cell disease and other kinds of hematological disorders like cancer. So I'm going to just summarize by saying that charge provides the stickiness or stealth that is really useful in targeting cells and tissues. Um, it can control trafficking, and it gives you this highly tunable surface chemistry that is very easily modified. And we're really interested in building these systems to target immune cells, stem cells, inflammation in general. And this is the work that we're doing now. But I'd like to describe really briefly how I got started working on these kinds of problems. I first want to mention that I am from Detroit, so I am a Michigander, grew up there a long time ago, and that I had, a great family. This is my older and younger brother, staggered evenly when we were small. My mother was really good at dressing us, and she was an RN and became the dean of a nursing college. Wayne County Community College nursing school was founded by my mother. And my father was a PhD biochemist, one of very few Black biochemists at the time. And he graduated from Wayne State University and became the director of health laboratories for the city of Detroit. So we were really encouraged to seek our education. And when I was growing up, I was really interested at first in being a writer of children's fiction. But when I got into high school, when I was about this age, I had my first chemistry lab, and I got really excited about how you can transform molecules from one to the other with simple steps. And just being able to do that in chemistry lab got me excited about chemistry because I was also very interested in math and physics. My teacher at the time told me to explore chemical engineering. Ultimately, I ended up going to MIT as an undergrad. I got my degree in 1984. And then later I went back to MIT to get my PhD in 1993. In between those years, when I graduated, I got married. I worked for 2 years in Fort Lauderdale, Florida, as a process engineer for Motorola. I then moved to Georgia Tech for 2 years, was working there at Georgia Tech Research Institute. I got a master's at Georgia Tech in chemical engineering there. And then I heard about a polymer program at MIT that sounded really exciting. And because I've become really interested in polymers and how you can change their structure and their property with just small changes, that I went back to MIT to study polymers and entered that program to get my degree. And during that time, I had a large number of mentors, and my mentors looked all different kinds of ways, coming from several different backgrounds, and all having different things to bring, which is something that was important for me. For example, my research thesis advisor, Michael Rubner, was a junior faculty member at the time and modeled to me how I could be a professor and still have a life. Um, Professor Bob Cohen was a big cheerleader for me and is the reason why the department head in my department asked me to interview. He was the one who sort of uh, let him know that I should be considered. Jim Gates was one of the few Black professors that I knew at the time at MIT, and was a great friend and colleague. A lot of the people that were my mentors were also my friends because you learn from your peers as well. Christine Grant, who was just a couple of years ahead of me at Georgia Tech, Christine Ortiz, who's a friend and collaborator here at MIT in material science, Gilda Berabino, another chemical engineer from Northeastern University, who ultimately went on to become dean and then president at Olin College. So I would say that some advice to you is to reach out to others at every point in your career because these are people who will have great influence over your life and provide you with critical advice and influence. Now, I also mentioned that I worked on polymers in the beginning. I was working on different kinds of polymers. I had an entire period of working on electro-optical and electrochemical applications, batteries and fuel cells. Then I did a sabbatical and at Caltech, and that was when I began to work on biology and biology-related material systems, and really began moving into different areas and fields. Um, in doing that, I had to go from being really experienced to entering a field where I was the inexperienced one again, but have my own ideas about what can and can't be done. And sometimes you get told that that idea will never work, or why are you using that approach when everybody just does this instead? So my advice is to not allow discouragement or critique to defeat you. You know, negative feedback is something that can be informative, can help you learn and grow. But it doesn't mean that you need to drop your own unique perspective. That is what makes you unique. That is what we need to see in the world because only you can bring that viewpoint and that perspective. All right, so I just want to finally say that it is the people that really matter, whatever career you choose to go on. I'm very lucky that I get to work with really smart, brilliant people, and these are some of the past students from my lab that have really had an impact in the world. I'm really proud of them. I think that they are even more important than, the work that we do in our lab because they're going to go out and do many great things. Um, I think about the fact that I am lucky to have like a research family, but it is also important to have other things in your life. Um, One of the biggest honors for me was being able to be on the President's Council of Advisors in Science and Technology for the Biden administration and be able to with a really interesting group of other scientists thinking about important problems that America has to address. Finally, I'd like to suggest you build a complete life because family really matters. Take time out of your life to honor that and to enjoy them. And vacation matters too, so take that time. I'd like to acknowledge those who did the work. I tried to highlight their names in bold for the talk that I gave today and the funding that helped to support it, and welcome any questions that you might have. So thanks. It was great to be able to talk with you. I'll now stop sharing so I can see people.
Okay, thank you so much for your presentation. I'll open up with the question I have. So with your research that you've seen a lot of progress with, do you see, like, foresee any clinical applications to it? And what would the process look like for like taking your research and then like doing some clinical trials with that?
Yes, that's a really great question. And in some of the work I just described with the ovarian cancer sticky nanoparticle, we're right in the middle of having those conversations. My former student graduated, but has stayed in the area to help work on the idea of a startup. And right now we got some seed funding to be able to do some of the key experiments. Some of those experiments are testing our nanoparticles in a lot of different cancer models. We had maybe two or three. We should look at five or six different kinds of models that replicate human cancer as best we can. Um, Second, we have been looking at manufacturing, how to generate these nanoparticles in a rapid and simple fashion. And my student has really found a great way to do that. Um, Third, we are looking at what kind of uh, what form of cancer we can address most readily. You know, patients are at many different stages, and for a trial, we have to understand what patients would be best for a cancer trial. Um, and now we actually have a few clinical collaborators in ovarian cancer to help us with that. And finally, it's looking for money. So my student is involved in a program that is helping to build that network, and he is building you know, his talk, his pitch for going out and fundraising for this effort.
So as a follow-up, then, do you work or seek collaborations with hospitals, with established cancer institutes? Because there are a number of them around the country.
Yes. In fact, in terms of cancer, we are working with the Dana-Farber Cancer Institute. We have a collaborator there, and so that is really helpful. But I'm also involved in something called Breakthrough Cancer. And in Breakthrough Cancer, um, it's a nonprofit that is meant to tie the cancer centers together. So there are five cancer centers working uh, with each other. And among those, we have a connection at UT Southwestern, um, and another one at Memorial Sloan Kettering. And so we're hoping we can leverage those two because it is important to be able to get a broad population. Maybe not for the first clinical trial, but in a broader, like phase 2 or 3 clinical trial. And I think that's going to help us a great deal. I don't know if that answered your question, Desmond.
Yeah, it did. It did. It does. Go ahead.
Yeah,
Might have
I was gonna ask how durable the nanoparticles are and if there's anything in the body that may degrade them over time.
Yes, they will degrade over time. Typically, the delivery time period that is relevant from the work we've been doing is usually over a period of maybe 1 to 4 days. We actually look at that, if you look at the dosing, it was a 7-day apart dosing. And what we have found is that when you first deliver the nanoparticle, that first polyl-glutamic layer, that outer layer, a little bit of it sheds so that you still have a net negative charge, but you have lost a little bit of the polymer even in the very beginning. But it's still sticky. It's still binding that amino acid transporter. So we get it sort of embedded on the cell membrane surface. In the first 24 hours, we know the IL-12 is still essentially co-located with the cell. Like you can do labeling and see that the the loci of the IL-12 label and the nanoparticles overlap. But over time, we are observing that this is for the maleimide system, the lipids are actually exchanging out of the nanoparticle and inserting into the cancer cell membrane. And so one of the reasons this works so well is because we are decorating over longer time frames the cancer cells themselves with this bound IL-12 molecule. It took a while for us to unwind to understand the depth of this mechanism. Initially, we thought we were just presenting from the nanoparticle. Then we did several experiments to demonstrate that if you design a liposomal system so that exchange cannot happen, you no longer get this sustained effect. So the short is it does degrade over time, and we take advantage of that in the formulation.
So could you tell us a little bit more about your work with glioblastoma?
Yes, in our work in glioblastoma, we actually use the design of this nanoparticle both to find a system that has a very specific affinity to glioma cells, and it follows the same kind of story that you saw with ovarian cancer, that we looked at an even bigger library and found these outer layers that have affinity for glioma. Then we wanted to find a system that had high transport through brain tissue. And the reason for this is that we were very interested in convection-enhanced delivery, which pushes the nanoparticle past the blood-brain barrier and allows it to diffuse through the tumor tissue. The trick, though, is that typically nanoparticles don't diffuse very far because they get caught in this sort of mesh of the tissue. So we found that by combining the sticky nanoparticle with a little bit of polyethylene glycol that helps it transport, that we're able to get a controlled diffusion through the tumor and selective uptake by glioma cells, greatly lowered uptake by astrocytes and neurons and other healthy cells. So that is where the net of our work in glioblastoma has gone. We have worked on nanoparticles that can go across the blood-brain barrier, and I find that also to be interesting, where you introduce it in the intravenously. And in that case, we've seen some success, but I would say the convection-enhanced delivery is the one that seems to give us the stronger and more robust positive response.
So that's the technological problem or challenge with glio, right? It's the blood-brain barrier. And so are you having to modify the surface of these nanoparticles to cross the blood-brain barrier?
Yes, we are. We are introducing peptides that have bind a specific set of receptors that are on the surface of the endothelial cells, the blood vessel cells that are unique to that blood-brain barrier, and allow transcytosis, or crossing the entire cell to the other side. There are a series of peptides that have already been published in literature, and we began to look at ones that had charge and used the charge to actually introduce these peptides onto the surface.
Okay, okay. It looks like Joel has a question.
Yes, Joel.
You could unmute, Joel.
Hello. Hi. Yeah, my name is Joel. I just wanted to thank you for your work on breast cancer. I know someone with breast cancer, so it just made it so much more real. Yeah, I'm taking a freshman level course here at Andrews, FB, Foundations of Biology, where we learned a little bit about cell signaling and a little bit about the immune system. So I wasn't completely lost because I haven't taken a chemistry class yet. So thank you for your explanation. It was simple enough for me to follow along. So just my first question is just why is it, because you mentioned about ovarian cancer, why is it that there aren't that many cytotoxic T cells there to attack the cancer cells?
Now, that's a great question. This has to do with the nature of cancer. Some of the most aggressive cancers undergo adaptations that allow either the immune response to be turned off, even though the immune cells are present, or in the case of ovarian cancer, glioblastoma, and there are certain other cancers, have essentially a matrix in which very few immune cells are even infiltrated. And there are a number of complex biological mechanisms that lead to this, what they call tumor microenvironment. And it's not really fully understood why tumors are so good at excluding these immune cells. So here at the institute I'm at, there are biologists whose primary area of research is understanding how tumors manipulate the immune environment around them. So it's a wonderful question and one that folks are studying even now. I see that we're getting close to the end, but I did want to address one of the comments from Jasmine. It says, family is really important in your life, huh? With all due respect. And hey, all due respect, yes, absolutely. I have a son who is very important to me. I was married. My husband passed away, unfortunately, a little while ago. But we had a wonderful life together, and I continue to have a wonderful life with the people who are important and close to me. And the way that I did that was by essentially very intentionally setting time aside for them. And that's the right reason why you see it in that slide, because my advice to you is to pursue what you want, but to be very, very deliberate in setting time aside for your family. Does that help, Jasmine?
I think it does. You want to?
Okay, thank you so much for speaking. I know you're on a tight schedule. And do you have anything to say though?
No, just we can't be like 30 years without seeing each other again.
I know, we cannot.
We
Absolutely.
cannot.
Oh,
Yes. Thank you so much for accepting our invitation and continued success, because we know it's benefiting a lot of people in the work you do. Thank you so much.
well, thank you. It was great talking with you all. You take care.
You too.
Hey,
Thank you very much.
yeah,
Bye-bye.
bye-bye.
Are you, will you not nodding off? I was seeing the head going like that sometimes. You need to go sleep. All right, thank you.